Today, let's learn about the fundamentals of chemistry for biology.
Now let's pause for a minute and talk about why we even care about chemistry for biology, because at first glance, chemistry can feel pretty far removed from bio. There's lots of atoms, bonds, and rules that seem abstract, but honestly, chemistry explains why biological systems work at all. So, one of the most important ideas we'll keep coming back to is this: biological function depends on chemical interactions. And chemical interactions are driven by chemical bonds, shape, and charge, which is why we talk about it. Let's start with a real world example first. This example is going to have to do with morphine and natural endorphins. So, your body naturally produces molecules called endorphins. These are signaling molecules that help reduce pain and create feelings of well being. Endorphins do their job by binding to specific receptor proteins on the surface of cells, especially nerve cells. So, here's a key idea: morphine and other opioid drugs only work because part of their chemical structure mimics the chemistry and exact shape of regions of natural endorphins. Think about these signaling molecules as little keys, and all of those receptor proteins are like locks. If it's a perfect fit, it sets off a chemical reaction that makes you, in this case, feel really good. So they aren't identical molecules, but they are chemically similar enough in certain key regions that the same receptor protein can bind them. From the receptor's perspective, it's not asking, like, is this a natural molecule? It's just asking, does this molecule have the right shape and chemical properties to bind here? That ability to bind depends on, once again, shape of the molecule, types of chemical bonds it can form, the charges and polarity of different regions. This is all stuff we're going to talk about today, and this is why chemistry matters. Now, this same exact concept can actually be applied sort of on the flip side. So you might have heard of Narcan. Chemistry name is naloxone. This is a drug that really saves lives. So Narcan is a life-saving opioid overdose drug. It works for the same chemical reason, binding, but it has a little bit of a different effect. So Narcan has a chemical structure that allows it to bind to the same opioid receptor proteins as morphine or heroin, right? So sounds similar, but here's the crucial difference. Narcan binds more strongly, and it also doesn't activate the receptor in the same way these other signal molecules do. So instead, it's just displacing all those opioid molecules. In other words, Narcan doesn't break the drug down, it doesn't destroy it, it simply just outcompetes it for the receptor, kicking out the opioid and legitimately saving lives. It means that breathing can resume because the receptor is no longer overstimulated. So this is chemistry with immediate biological and human consequences. So let's rewind and start with the basics. Way back, Aristotle decreed that there were four earthly elements: air, wind, fire, earth. This is 4th century BCE. This framework actually dominated Western thinking for like 2,000 years until it was gradually replaced by the scientific revolution in the 16th and 17th centuries by experimental chemistry and atomic theory. So the French chemist Antoine Lavoisier was one of the first, most important figures of that time. He performed a series of experiments that made him realize that there was these simple substances that he would later call elements. Nitrogen, hydrogen, oxygen, carbon were among some of the early ones that he classified. The cool thing is, if you fast forward 87 years to around 1869, when this Russian chemist Dmitry Mendeleev started working on this, he arranged 52 known elements into rows and columns, and that's how we got the periodic table. The really cool thing about it is that he organized them based off of atomic mass and chemical properties and left some gaps for things that hadn't been discovered yet. But because of this organization, he actually accurately predicted their properties based on where the gaps were in the table. And this was a really major turning point in chemistry because it showed that the properties of elements followed a predictable pattern, paving the way for modern periodic table.
In biology, we tend to focus on 4 particular elements most: hydrogen, carbon, nitrogen, and oxygen. And that's because these 4 elements together make up about 96% of living matter. So 96% of your body is basically made up of just these 4 elements, and that's why they get so much attention. Now, what makes an element unique? We're going to be talking about atoms and elements, and it's nice to know how they're similar and different. Think about it for a second. So how would we define helium versus hydrogen, right? What makes them different? So you might be thinking they have different quantities of subatomic particles? Yes, but it really actually just comes down to one. So the number of protons is what makes an element unique. For example, hydrogen just has one proton, whereas helium has two protons. When we talk about atoms, this is the smallest unit of matter that still retains properties of an element. So I just told you that an element is defined as the number of protons that the atom contains. You can have carbon-14 versus carbon-12. These are different types of atoms of carbon, meaning that they are carbon, they have that identity, they have the same number of protons, but we give them different names because they might have different numbers of neutrons, for example. Or we might have an ion of a certain element, meaning it's still that element, it still has the right number of protons, but perhaps there's a differing number in electrons, and so we get a certain charge. We'll get there if that doesn't make sense yet. For now, let's start by drawing an atom. So here I have an atom that has 2 protons, 2 neutrons, and 2 electrons. So when we're drawing these out, there's various ways to make models. We're going to stick with a simple version for the sake of just intro biology. In the middle of an atom are the protons, and protons have a positive charge. That's why I'm putting a plus sign. This is also where you find the neutrons, which are thought to have a neutral charge. And then we have electrons that circle and move around that center. There can be various levels of electrons, and the first level only holds 2. So I draw my first electron and my second electron here, and there we have it, our example atom. And it contains all of the subatomic particles. So think sub means below, so subatomic is what makes up an atom. We have electrons, protons, and neutrons. Now the notation that you'll often see is a letter for an element. So for example, carbon is referred to with a C. And then in the top left corner will be the atomic number. The atomic number is the number of protons. And I just told you number of protons is like identity of the element. So think about that when you're trying to remember what atomic number means. It's the number that determines what atom or element we're looking at. And then the mass number is taking into account protons and neutrons. You might be thinking, what about the electrons, right? If we're thinking about the whole mass of the atom. For the sake of biology, just think of it as electrons are so tiny that they barely take up any space or mass at all when it comes to the atom. So we just kind of take into account the protons and neutrons, and we're going to call it good there. So we got 2 practice questions here. So the first, hydrogen contains 1 proton, no neutrons, and 1 electron. Given this information, practice writing the shorthand notation for hydrogen using the format that we're seeing here. So pause the video and try this out. Every time you try it out on your own, it sticks even better, even if we're feeling a little lazy and we don't want to do it. So I'm going to start writing it out. Make sure you pause and give yourself a chance. H is for hydrogen. You don't have to have this memorized. Go ahead and look up a periodic table. You can always find the element. And then in the left-hand corner, top corner, I want to put atomic mass. So I'm told here that there's just 1 proton. For the mass number, I look at protons and neutrons. I was just told that hydrogen actually doesn't have any neutrons. So my mass number here is going to be the same as my atomic number. The second is, how do we draw an atom of hydrogen? So just like we did in the last example, I start in the middle. I know I have 1 proton. There's no neutrons, so that's it for the center. And then I have 1 electron circling that center proton. As a side note, as we're moving forward in chemistry, you're going to see H+. This is the hydrogen ion. An ion is when we have an atom that has a certain charge to it. So the hydrogen ion is when this hydrogen atom, which usually has a proton and an electron, it loses its electron. If it loses its electron, we just have 1 proton on its own. This is something we will return to, but I wanted to bring it up while we're talking about hydrogen here. Whenever you see H+ or hydrogen ions, that's the same as referring to a single proton by itself. So here's a nicer drawing showing the arrangement of our subatomic particles in an atom. When we have atoms that have a neutral charge, we can always just assume that the protons and the electrons are in the same quantity. If I've got 2 protons, I've got 2 electrons because it's neutral, so it's balanced out. The ions, which I just brought up on the last page, that's when we have an imbalance. So perhaps there are more protons than there are electrons, or perhaps there's an atom that picked up an extra electron, so it has an overall negative charge. I'm going to repeat that. If there's more electrons than there are protons, we have an overall negative charge because electrons are associated with negative. If we have more protons than we have electrons, then we have an overall positive charge. And these are ions. For now, though, just know if there's a neutral atom and you know that it has 6 protons, it also has 6 electrons. Okay, so an atom of sodium, Na, is depicted in the figure below. In this figure, sodium is not ionized, meaning that it's neutral. It has no charge. Given this information, how many protons does sodium have and how many neutrons does it have? Pause the video, think about this, see if you can get the answer. Okay, I'm going to walk us through it. So I'm given some information here. I know that the amount of protons and electrons should be the same because we're not charged. I'm also given a mass number, and I know the amount of electrons because I can just count them up here. So what I'm going to do is I'm going to count the electrons first. I have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. Okay, so I have 11 electrons. That means I also have 11 protons. And I know that mass number is protons and neutrons. So now we just plug in what we're missing. We're told that the mass number is 23. So 23 equals 11 plus neutrons. I know I have 11 protons already, so actually that even just crosses out D and it crosses out B. And so how many neutrons do I need to get to 23? It's going to be 12, and that's how I get at my answer, C here. So it seems kind of intimidating at first, but it's actually some simple math using the equations that we just learned about. Let's do some other practices. So chlorine can form 2 main isotopes. We haven't learned the word isotope yet, but remember how I brought up that you can have different atoms for a given element. They have the same number of protons always because that's the identity of an atom, or of an element, sorry, but they can differ in those other subatomic particles. So here I have I have 2 versions of chlorine. The atomic number of chlorine is 17. You should already be trying to translate this in your head. What does that mean? If the atomic number is 17, that means chlorine has 17 protons. Question number 1 says, 1 isotope of chlorine has 18 neutrons. What would the mass number of this isotope be? I know that mass number. I'm going to write it out for us. It's protons plus neutrons. I'm looking for mass number. I know that we have 17 protons and 18 neutrons. So what's 17 plus 18? I'm gonna get 35. Another isotope of chlorine has a mass number of 37. How many neutrons does it have? So I just figure out what's missing here. I don't know neutrons. I do know 37. I know 17. So if I subtract 17 from each side, we get 20 neutrons.
Let's pause to think about why we care about isotopes at all. Here's a cool example. So some isotopes let us measure time in biology and Earth's history, something we really couldn't do otherwise. Great example is radiocarbon dating. So I'm going to oversimplify here, but carbon exists in different isotopic forms. There's carbon-12, carbon-13. These are really stable. Carbon-14 is radioactive, meaning it breaks down over time. While an organism is alive, it's constantly taking in carbon from the environment through food, water, the atmosphere. If it's a plant, it's taking in carbon dioxide, and then, of course, animals eat plants and we receive that carbon. So, the ratio of carbon-14 to carbon-12 in a living organism closely matches the ratio found in the atmosphere. But once an organism dies, that changes. So, no new carbon is taken in, and the carbon-14 that's already there begins to decay into nitrogen-14 at a known, predictable rate. The carbon-12 doesn't decay, it stays put. So by comparing how much carbon-14 remains relative to the carbon-12, scientists can estimate how long it's been since that organism died. This is pretty cool. It means that tiny differences in atomic structure give us a biological clock, one that lets us date fossils and archaeological remains.
The outer shell of an atom is called the valence shell. We know that there can be various electron shells. There can be 1, there can be 2, there can be 3 electron shells. The valence is not any one of those in particular. It's all about context. So it's always the outer shell, no matter how many shells there are. For helium, the valence shell is the first because there's only one shell. For fluorine, here in group 17, the valence shell is the second shell because it has 2 electron shells. An atom is most stable when the valence shell is completely full. Think of it as like the atoms are happy when their valence shell is full, they're stable, they're less reactive. So another way to say this is that the lowest potential energy for a valence shell is with full 8 electrons, especially if we're talking about that second shell or the third shell. The first electron shell only holds 2 electrons, and then the second shell holds 8, and the third shell holds another 8. In this introductory biology, we're not going to get much past that, so you can just focus on those first 3 shells, 2, 8, and 8. So let's say we have some different atoms, and they don't have their valence shell full. Well, a couple of things can happen. An atom can steal an electron from a different atom and fill its electron shell. Or I should say, its valence shell. That's what we're seeing here. So there's 7 electrons in the outer valence shell for this first atom here. And if it were to gain 1 more, that outer shell would be full and this would be more stable. On the example under it, we have an atom with 3 electrons. So the first shell is full with 2. The second shell only has 1. It would need to gain 7 electrons, which is unlikely. It's not going to happen. Instead, the easiest thing for this atom to do is get rid of an electron. So let's draw it together. This would presumably have 3 protons in the center, and then 1, 2 electrons, and that's it. It gave away the electron in its second shell to become more stable here. Another strategy besides giving away and taking is to share. So here we're seeing 2 oxygen atoms. And we're only looking at that outer shell, by the way. So oxygen has 1, 2, 3, 4, 5, 6, 7, 8 electrons total. It has 2 in the first shell and then 6 in the second shell. That means there's 2 spots, 1, 2, where oxygen is missing its electrons. And we often draw these electrons in pairs. So what we're seeing here is these 2 oxygens are teaming up and they share their electrons. When they do that, they are essentially filling their outer shell, right? If you focus on this first oxygen here and take into account the electrons it has and the one it's sharing, well, it now has 8 electrons in its outer shell. It's stable. If we look at the oxygen on the other side and take into account the one that it's sharing, it has 8 electrons on its outer shell. Again, this drawing isn't showing the inner shell. So you'll often see that drawn as oxygen bonded to an oxygen. And the reason that there's 2 lines instead of 1 is this is a double bond. It's bonding in 2 spots. We often can instead see atoms just sharing 1 pair of electrons, but in this case, it's sharing 2 pairs of electrons. So in summary, atoms want to fill their valence shells. This is what explains reactivity in a lot of chemistry and chemical reactions. We can do that by giving away electrons, taking electrons, or sharing them. So now let's think about some of these chemical bonds. And we sneakily already introduced them. So the ionic bond, this is that scenario where we have a giving away or a taking of electrons. So you can call this a transfer. Sometimes it's referred to as like a stealing or giving away. But these atoms are going to lose or gain electrons. Here we have NaCl. It might sound familiar. Sodium chloride. NaCl is table salt. So sodium, and I actually want to point out too, this diagram once again is just showing the outer shell of these atoms. So there are more electrons here. In sodium's outer shell, it just has 1 single electron. It's easier for sodium to get rid of that electron than it is to take on a whole nother 7. So it wants to get rid of that so it can become more stable. Chlorine is the perfect match because in chlorine's outer shell, it has 7 electrons and it needs 1 more to be stable. So they're pairing up. Sodium gives away its electron, chlorine accepts it, and now we have an ionic bond. Ionic bonds are kind of interesting because they are very strong in a solid state. Think about the salt crystals, on your kitchen counter. They're literally crystals, they're hard. But you put that in water and this bond becomes very weak and it falls apart. Covalent bonds is the name for when we have sharing of electrons. So when atoms are coming together through covalent bonds, this is molecules. So here we have an oxygen and 2 hydrogens. You should be recognizing what this compound is. 2 hydrogens and 1 oxygen, that's H2O. The substance of life, that is water. So if we look at our oxygen here, once again, just seeing the outer shell, oxygen needs 2 electrons to be stable. Hydrogen has 1 electron when it's neutral. If it were to gain a second electron, that first shell would now be full and stable. So what we do is we team up. This hydrogen shares with the oxygen, and this hydrogen shares electrons with the oxygen, and now everybody's happy. You can really clearly see how oxygen's outer shell now has 8 electrons and how hydrogen's outer shells each have 2 electrons. Their valence shells are both full. And we write that out like so. The hydrogen bonded to an oxygen, bonded to another hydrogen. Here we have 1 single line showing 1 pair of electrons being shared versus that double line that we saw when it was 2 oxygens together. As we talked about before, you can have 1 pair of electrons shared, denoted by 1 line, or here 2 pairs of electrons are being shared, and that's a double bond that we see with oxygen. Either way, it's a covalent bond. It's a sharing of electrons. So we mentioned valence, and you probably caught on to this by now, but valence helps explain how many bonds an atom is willing to make. So valence itself, the word alone, this is a tricky one, okay? The word valence is the number of unpaired electrons in the outer shell. And I'm sure some of you are like, I thought you told me it just was the outer shell. So this is different from valence shell. If you say valence shell together and like as one unit, the valence shell is the outermost shell, the end. But if I'm talking about valence, the word on its own, that's referring to the number of unpaired electrons. So draw an atom of carbon. How many bonds can it make? To help you out here, carbon has an atomic number of 6. Pause the video, draw it out on your own first. And I'm going to start drawing it. So carbon is going to have 6 protons. I'm just going to write in 6 protons, and that's going to be my center. I'm not really worrying about neutrons right now. And then in the first electron shell, I can only hold 2 electrons. So I go 1, 2, but I haven't gotten to 6 electrons yet, so we're moving out to our second shell. So 1, 2, 3, 4, 5, 6, and now I stop because I've reached 6 electrons, and this is a neutral atom. So if I'm trying to see how many unpaired electrons do I have, there's a spot next to each electron where we could make a pair. Another way of thinking about this is how many electrons does this atom need to fill its valence shell? Well, it has 4, it needs 8, so 4. The valence here is 4. And it also explains why carbon can make 4 covalent bonds. Let's try this on a different one. So we have an atom of nitrogen 13 shown below. How many bonds can nitrogen make? Pause, think about it on your own. It has 7 protons, 7 electrons, and 6 neutrons. Really, the only important thing that we want to be focusing on to answer this question is how many electrons it has. So there's 1, 2 in the first shell, 3, 4, 5, 6, 7. What I'm seeing here is 1 pair of electrons, and then 1, 2, 3 that have an opportunity to bond covalently with something else. Again, another way of thinking about this is how many more electrons would we need to fill that outer shell? Well, we would need 3. So nitrogen can make 3 bonds, covalent bonds in this case. Now we're going to get a little bit more into the nitty-gritty. So when it comes to covalent bonds, there's actually 2 types, polar and non-polar. And all of this has so much application further into biology. To understand polarity, we have to take a minute to talk about electronegativity.
Electronegativity is a measure of how strongly an atom pulls on electrons when it forms a bond. In other words, how badly does this atom want electrons? This matters in biology because electronegativity helps determine whether bonds are polar or nonpolar, which affects things like molecular shape, solubility in water, how molecules interact with proteins and membranes, etc. We'll get there later. So if you look at the periodic table, electronegativity is going to follow 2 main trends. Number 1, it increases as you move up a column. That's because atoms near the top of the periodic table hold on to their electrons more tightly. They have fewer electron shells, and their nucleus is closer to the bonding electrons. Basically, it's just a shorter distance, so that means a stronger pull between the protons at the center of the atom and the electrons surrounding. So just think, they're physically closer together. The second trend is that it increases, or electronegativity increases, as you move to the right across a row. As you move to the right, atoms have more protons in their nucleus. Think of it as just like this stronger, bigger positive charge that's going to be pulling on the electrons. So atoms on the right side of the periodic table tend to be better at attracting electrons than those on the left. And you put those 2 trends together and the electronegativity increases towards the top right corner of the periodic table. You might be noticing, though, this grayed out area, there's one important exception. This is the noble gases. Even though they're on the far right corner, noble gases generally don't form bonds. So electronegativity values usually aren't even assigned to them because electronegativity only really matters when atoms are sharing electrons in a bond, and the noble gases, they don't really do that.
So in a polar covalent bond, this means that there is an unequal sharing of electrons. So take for an example, water. Oxygen is one of the most electronegative atoms. It has a tendency to pull electrons towards it. I think of it as literally like a little game of tug of war, and the oxygen is really, really strong, and it's kind of hogging all of those electrons. So it ends up with, let's say it was like really hogging all of the electrons. It had 2 electrons in the first shell, 8 electrons in the second shell. So we're working with like, like almost 10 electrons. Meanwhile, in the center, it has the same number of protons it had before. So overall, there is a negative partial charge because we're kind of like feeling the effect of all those electrons being hogged towards oxygen. And that partial charge is often written, if we were to zoom in here on this spot, in this Greek symbol with either a negative or a positive sign, a partial charge, partial negative charge in this case. The hydrogens, I like to imagine, they were only one proton and one electron to start. The electrons being like pulled away from it, so I'm feeling the effects of that lone proton. So they end up with partial positive charges. And that's what a polar molecule is. It's a molecule that is made up of atoms that are covalently bonding. They're sharing electrons, but at least one of those atoms is way more electronegative than the others, and it's hogging electrons and creating partial charges on the molecule. So that was our polar bonds. How about non-polar covalent bonds, aka equal sharing? When is this going to happen, considering our four main elements that we focus on in biology: carbon, hydrogen, nitrogen, oxygen? So the first case is whenever we have an element bonded to itself. So a great example of this was atmospheric oxygen, O2, oxygen bonded to another oxygen. This is like a game of tug of war where you have a clone of somebody. So someone's up against themselves, or two people, same size and weight, right? They have the same strength. You have a really even game of tug of war on those electrons. You're not going to get partial charges. The second is more of an exception, where we have, anytime we have carbon bonded to hydrogen, and this is super important to remember. It really helps us understand the biological macromolecules later on. So the reason this happens is because these are different atoms, but they have similar enough electronegativities that their pull on the electrons is it's equal enough that we don't get partial charges. And then there are some molecules where you'd think they'd be polar because there's an element that's more electronegative. For example, for carbon dioxide, CO2, oxygen is more electronegative than carbon. This, maybe would create some sort of polarity, but the exception is because of the way that the atom is configured. Let me just show you what I mean. So in carbon dioxide, I have an oxygen double bonded to a carbon, double bonded to another oxygen. It's set up in a way that is perfectly symmetrical. So each oxygen on either side of that carbon is pulling on the electrons equally, and it ends up creating this symmetrical scenario where we don't really have partial charges. So we can find more exceptions out there in chemistry, but this is really the only biologically relevant one that we focus on. So just keep in mind these three rules for when something is going to be non-polar. And then you can just kind of assume that everything else will be polar. So something's going to be non-polar or equal sharing when we have an element bonded to itself because they have equal strength. Carbons and hydrogens bonded together, super important. And then we can think of carbon dioxide as just one little exception. So for example, at this same table, if I take a look at the bottom two, we have two different examples of nonpolar molecules. The bottom one is carbon dioxide, and that's just because of the shape of the molecule and that equal pull of the oxygens, even though carbon is more electronegative. On the top here, we have carbons bonded to hydrogens, and that was the really biologically important exception I wanted you to think about. Anytime we have carbon bonded to hydrogen, think hydrocarbons, think nonpolar. So both the bond type and molecular shape, in the case of carbon dioxide, are going to determine whether a molecule is polar or nonpolar. Now, a question I often get is, how do I know if an atom is going to ionic bond or participate in a covalent bond? A general rule of thumb is if the atom has only like 1 or 2 electrons that it would need to lose or gain, it's probably more likely to participate in an ionic bond. If it's something like carbon, it would have to gain 4 electrons or lose 4 electrons. That number is getting kind of high, way more likely to participate in covalent bonding and sharing. So, to get this all straight, I would pause, see what you can fill out on your own. But what's really helpful here is it gives us a moment to think about the strength of these bonds. So the first one I have here is the polar covalent. We learned that this is unequal sharing. Versus nonpolar covalent is equal sharing. It's equal sharing, so we get no partial charges, and so the molecule's not polar. These are both considered strong bonds. They are the glue that hold together molecules. And actually, once they're formed, if you want to break those bonds, you have to use energy. It requires energy to break those bonds. They're just so strong. Ionic bonds, in a solid state, they are relatively strong, but in water, they are weak. And this is important when we think about acids and bases and why sometimes in chemical reactions you have a certain compound or molecule, you put it in water and it dissociates, meaning the ionic bonds fall apart. And then we have hydrogen bonds. We haven't talked about these yet, and I kind of want to keep them separate because hydrogen bonds are not truly in the same category as our ionic and covalent bonds. Ionic and covalent bonds tell us what's happening between atoms in molecules or compounds. Hydrogen bonds happen between molecules, usually. And they are weak interactions. So right, comparing to covalent bonds, a covalent bond is within a molecule. Hydrogen bond is between molecules. Super important distinction. Oh, and let's just round us off here. Sharing, and let's just round ourselves off here. This is a transfer of electrons. So here's an example of hydrogen bonds. All of those red circles, those are oxygens. All of the white circles, those are hydrogens. This is just another way to visualize atoms, molecules. So what I'm circling right here for an example, this is one water molecule, and it's held together by covalent bonds. There's 5 water molecules here, and between those water molecules, that's where we get hydrogen bonding. Because remember, water molecules are polar, meaning that there's partial charges. There's a partial negative charge on the oxygen and partial positive charges on those hydrogens. And opposite charges attract. So the partial positive on one water molecule is going to be attracted to the partial negative on another water molecule. And this right here is huge because it really helps explain why water is foundational and essential to life. We're going to talk about that in another video, but kind of incredibly, it actually comes back to water's ability to form hydrogen bonds. So to give you a sneak preview, it's one reason why water is so good at being a solvent, meaning that things dissolve in it. So think about your coffee in the morning, you put a sugar cube in there, it disappears, it dissolved in water. It's why we are largely just big bags of water with like other components. Water is the solvent of life. And it's because it can hydrogen bond with other molecules, like here our salt. I told you that ionic bonds are really weak in water. So NaCl, a salt put into water, gets ripped apart. And that's because these water molecules are forming what's called a hydration sphere, and essentially hydrogen bonding all around those ions and separating them completely. Hydrogen bonds also occur in molecules of DNA. So you've probably at this point seen images of DNA as this double helix ladder. Well, that double helix is actually made up of 2 sides. Let's see, so if we were to flatten out our double helix, it would look like a ladder, right? If we unwound it. And then what I'm trying to say is that in reality, there's two sides to this ladder. And another side. Each side of the ladder is held together by strong covalent bonds. However, the two sides come together through hydrogen bonding. And we might not see it yet because we haven't learned about DNA, but this little key right here, the fact that they are hydrogen bonded together and we can easily unzip them apart, explains so much about genetics and how gene expression and other mysteries of the genetic world work. So we will be coming back to this. You're going to want to remember, oh yeah, hydrogen bonds. It really helps. It's a structure that completely correlates with the function of this molecule. So pause here, let's get a little practice in. Which bonds within this big molecule here would you predict are polar? I circled them and labeled one of these covalent bonds between a nitrogen and a hydrogen, A. There's a double bond between a carbon and an oxygen, that's B. And then a single bond between an oxygen and a hydrogen. You should have paused the video, so I'm going to give you the answer here. Which ones are polar? Well, nitrogen to hydrogen, carbon to oxygen, oxygen to hydrogen, these are all polar bonds. And I know really quickly because I don't see any of my exceptions. The exceptions I'm looking for is something bonded to itself, so like oxygen to oxygen. We're not seeing that. Carbon bonded to a hydrogen was my other exception. I don't see that. And those were the only two relevant ones here. Yeah, the rest of these have different electronegativities and they are polar. Here's another one for you to try out on your own. This can be a little bit tricky, but we have different atoms here and you're trying to figure out which one of them could form a cation. Pause. That means positively charged ion with a charge of +1. So pause and try to think this one through. Okay, so let's start at the top. For A, we are likely to see this electron leave because this atom would just need to lose an electron in order to have the stable 2. We're assuming that all of these atoms here are neutral. So if I'm seeing 3 electrons, I know that there are 3 protons, right? If I lose an electron, I have 3 protons, and then this would become 2 electrons. So I would have 1 more proton than I do electron if this ionized, meaning I would have a charge of +1. People often, when they do this practice problem, will look at D and think, oh, I just need to gain 1 electron right here. But that would give you a charge of -1 because you just gained a negatively charged electron. Last thing I want to talk about in this video is laying down the groundwork for talking about chemical reactions. Maybe you've seen them before, but they are written out as compounds or molecules on either side of an arrow. So if we are reading left to right here, I have my reactants that create products. Right here, there's just one reactant being shown, but in reality, you can have multiple different molecules coming together. I read this left to right now where I can say, okay, my reactants, hydrogen peroxide, can dissociate into water and oxygen. And those are my products of the reaction when it's progressing in that direction. So reactants are substances at the beginning of the reaction. Products are substances that form at the end of the reaction. And chemical reactions can be reversible or irreversible. If there's an arrow going in one direction, it means it's irreversible. So hydrogen peroxide will become water and oxygen, and it can't turn back into hydrogen peroxide. On the other hand, we have here HCO3 minus. This is bicarbonate reacting with a hydrogen ion to form carbonic acid. The double arrow tells me I can read this in either direction. So in one scenario, bicarbonate and hydrogen are my reactants, and they make the product carbonic acid. However, if I'm reading this right to left, I could say that carbonic acid, when it dissociates into these two ions, carbonic acid would be my reactant and it would make these products. So reactants versus products, it just depends on which way you are reading a reaction if it's reversible. Lastly, there are different functional groups in chemistry that we see a lot in biology, and it can be really helpful to recognize them and understand how they usually react or respond in chemical reactions. You don't need to memorize all of them per se, but getting familiar with some of them can help you later on. So these functional groups are groups of atoms within a molecule that can confer consistent, specific properties to these molecules. They often interact with each other or other functional groups via hydrogen bonds, and they each have different characteristics. So again, taking a moment to review them and start to get familiar with them so you can recognize them later could be worth your time. So thanks for joining me on this big foray into the basics of chemistry for biology. This should give you the foundation that you need as you move forward in your biology career.